Hello, I'm Rosaly Lopes, and I'm the president of the Planetary Science section. With me is Michael Mischna, the president-elect. And today we have the pleasure of having Dr. Penelope Boston, Penny, as our Sagan speaker. Penny is a senior advisor on science integration at NASA Ames Research Center. She's a geomicrobiologist and an astrobiologist. And she's going to present the Carl Sagan Lecture this year. This lecture is presented every year at the Fall AGU and recognizes a scientist who truly embodies Carl Sagan's interest in astrobiology, as well as his effective communication skills. It's hosted in alternative years by the Biosciences section and the Planetary section. So this lecture is to honor Carl Sagan, and after the lecture, I'll present Penny with a certificate. But now, my pleasure to welcome Penny Boston. Thank you.
It's great to see everybody here this morning. I actually knew Carl and miss him a lot still to this day, even though he's been gone from us for more than 20 years. Once published a paper with him, with Carol Stoker as first author, and interacted with him a lot when I was a graduate student and a postdoc. And so having had the benefit of that, of course, I was thrilled when I was invited to give this lecture. And thinking about what I would actually try to say, it's very difficult for a lecture like this. I mean, it's not, our last batch of experiments kind of thing, right? It's what have we learned from the number of decades now that we've been really engaged in a serious effort to look for all of the components of astrobiology, what we now call astrobiology. So I think that what I settled on was really trying to give a little flavor of some of the history of it, but also trying to poke at what we're doing now, of course using some of my own work as an illustration, but really not focusing heavily on that. And then thinking about some of the deeper questions that I think are very hard for us to tackle in our daily investigations and as we plan for missions and things of that sort. So it's been a very unpleasant week. I want to give a tribute to viruses because they are amazing little entities. And I had norovirus this past week, and I've been desperately ill. But it reminds me that the life forms that we're looking for on other planets, the way we're trying to understand our own evolution on this planet, is highly integrated with the history of viruses and other tiny components that are not cells but carry genetic material around. And so there are profound influences, and I have just lived through a wonderful experience of reminding me of that. So I'm a little shaky and weak today, but I'm at least standing up. So a tribute to the viruses, and I want to point out that on Friday from 8 to 12, there is a geovirology session. So if you're still here on Friday, I would encourage you to go see that, because astrovirology is a small field but growing, and one of my passions, not as a practitioner but as a fan. So astrobiology is a tremendously complex field with the vast array of different fields all pouring into it. There are clearly pieces missing. I ran out of place for circles up here. As we go further into the future, clearly artificial intelligence and machine learning are going to greatly facilitate our ability to conduct experimental work on far distant objects in our solar system, but also even here on Earth, our ability to integrate vast quantities of data, which all of these fields, as they pertain to astrobiology, are producing, is something that is a huge challenge for us in our future. We are paying attention to curating things and keeping databases available and so forth. But truly, I think the scope of astrobiology is so spectacularly across the disciplines in all different directions and across the cultures that go with those disciplines that we need considerably sophisticated help in trying to integrate all that so that we can make sense out of the things that we are all individually working on. So in spite of the fact that the last number of decades have been the time in which astrobiology has come into its own, we certainly didn't invent it a few decades ago. People have been thinking about these ideas for a very long time, and one of the earliest people to really come up with the idea of astrobiology and in fact coin the term, albeit in Russian, astrobiology and astrobotany. And this was a guy named Gavril Tikhov, and he was an astrophysics person and highly influential in promoting within the then Soviet part of the world these ideas of the legitimacy of looking for organisms on other planets. And I put in this one quote that I found on David Darling's excellent history website. Mostly, they took young graduate students. They carried out many studies of optical characteristics of terrestrial plants, including expeditions to regions with extreme climatic conditions, et cetera, et cetera. The object of these studies was to discover how low temperature, high altitude regime, and oxygen deficiency affected the optical spectra of plants. Well, this is the prototype for what a large number of us have been engaged in for many years, and that is seeking out places on Earth where there are conditions that may resemble some of the conditions that we find on other planets, and piece by piece in this puzzle put together some way of tackling how we think about life forms that we want to go look for, but we don't even know exist. Because this is a fine line between going across into science fiction or staying within the science realm but being imaginative so that we can have open enough minds to actually understand that we are finding life processes if we get into the position where we actually are so fortunate as to do that. This work of Tikhov had a great deal of influence. I show the cover of the actual first book, Astrobiology and the Ethics of New Science by Charlie Cockell, has a picture of Tikhov's first edition of Astrobotany, and this has still influenced us today. So I showed just the heading of a very important paper by Sarah Seeger and colleagues a number of years ago, I think it was 2005, on the vegetation red edge. Well, this is an intellectual child of the work that Tikhov was pioneering. Famously, Joshua Lederberg, who was an extremely brilliant Nobel laureate, receiving his laureate for work in genetics along with several other colleagues at the age of 33. So he was definitely a deep thinker and a far-ranging thinker. And he coined the term exobiology in the late 50s and wrote the sort of seminal flag-planting paper on that, which was published in Science back in 1960. And so that word exobiology got into our lexicon, and when I was a student, it was exobiology that I read about in my weekly reader as a 7-year-old and in school, and fell in love with. And at the same time, I fell in love with SETI, which the Drake equation, Frank Drake had written a 2-paragraph thing for this little kid newspaper. So in the spirit of Carl and Frank and all these other folks, astrobiology, exobiology has found its way into the public consciousness through the efforts of our predecessors and now through our efforts and well into the future. And it's a part of science that really, as I'm sure you all are well aware, grips the imagination of people who have very little patience for other types of science. So we have a unique leverage point within astrobiology to really get the word out there about not only astrobiology, but also understanding about how all these other sciences feed into it. And this is a very important message to get across to our broader public and certainly to young folks of all ages. And you never have to sell a second grader or a fourth grader on how wonderful space is. So it's very easy. Thinking about other terms that have been used, when I was a kid, I was an early science fiction reader, and one of the juvenile science fictions produced by the famous and renowned science fiction writer Robert Heinlein was The Star Beast, and I read that in the 60s. And it was this wonderful story about this time in the future when there were comings and goings with other planets and other star systems, and we had discovered all kinds of wonderful aliens of both intelligent and organic levels. And so this kid has a pet, and it turns out that the pet, sorry for the spoiler, is actually a baby of its species, of a very long-lived intelligent species, and it thinks that it's keeping him as a pet. So if you have never read this, I would suggest you go find a copy and read it, because it's a great read. So this term xenobiology that Heinlein coined in this book is now used in a much more restricted sense, and we are now applying it to where we are looking at non-naturally occurring chemistry, so alternative DNAs or biopolymer-like things that are not the natural biopolymers that we find in the natural world. And it's increasingly being applied to synthetic biology. And of course, even though synthetic biology is its own field, obviously there are huge overlaps between synthetic biology, between astrobiology, and in fact space biology, where we're looking at the fate and development of organisms from Earth that we actually put into a space environment. Even Winston Churchill got into the act. He was not an ordinary politician, I'm sure you are well aware of that, but of the various essays that he wrote one in 1939, which he never published, but revisited multiple times through the rest of his life on Are We Alone in Space? He revised it and then called it Are We Alone in the Universe? And this was his take as a statesman on what was the prospects for other life forms in our solar system and beyond. And of course, he explores a number of aspects of this, including what does that do to us, which we would now call astrosociology, right? So that was very prescient, and Mario Livio had a great paper out on that in Nature a couple of years ago. Cosmo Biology is something that people sometimes suggest to us that we rename astrobiology, and so I have taken pains to look it up, and it has a very mixed lexicon. Apparently, cosmobiology is used to apply to a particular branch of astrology. So this is kind of a direction that we probably are not very interested in moving into. It's also, much to my amazement, applied to a branch of medicine which makes more sense to me, where you're studying the effects of cosmic rays and other geomagnetic phenomena effects on humans. And so these are demographic studies and epidemiological studies looking at high sunspot and low sunspot and mass ejection, coronal mass ejection events, and what that does to various health indicators in the population. It has been used once in a single paper to refer to what we would consider truly origins of life, prebiotic experiments, which puts it in the astrobiology realm. So I think that's a term that we should stay away from. One of the other great themes of the 20th century that fed into our astrobiology is biospherics. And this idea of the biosphere was coined by a geologist named Edward Suess in his 1885 book, The Face of the Earth. And he mentions this, but years later, Vladimir Vernadsky in the Soviet Union actually took this idea up and ran with it. And because of the geopolitical situation of the time, it took a long time for his book, The Biosphere, to be translated into English and actually percolate into our consciousness across the ocean and in Western Europe and so forth. But that was a very important body of thinking that really has funneled into this. And so this idea of biospherics which eventually has come to mean the science and technology of analogs and models of Earth's biosphere, and particularly, of course, artificial Earth-like biospheres as a research tool. And this was the most official-looking body I could find for an official kind of definition. But I really think that we could extend this because as we begin to discover, as I'm convinced that we will, evidences of actual biosphere activity on exoplanets into our future, we're going to have opening before us the way to actually do population studies in a way that we cannot do very easily within our own solar system because we have only one set of planets, and to date we have only one inhabited planet to our knowledge. So I think there are many other ways you could make this up: cosmoecology, galactobiology. You know how in biology we love to make up these goofy names. But I think that astrobiology is really the preferred term still, and I wasn't sure about that when I started thinking about this. It is broader in its nuances than exobiology was. Exo meaning outer or outside, the biology of outside really was focused on how do we go to Mars and look for life, how do we go somewhere else and look for life. But really astrobiology has grown into this field that encompasses very deep time studies of our own Earth, deep questions about the origins of life on Earth and its early development. And of course we then export that in our thinking to the extraterrestrial context, but it has caused us, perhaps unforeseeingly, to delve even more deeply, I think, into our thinking about Earth than we would have if astrobiology had not arisen. And I think this shows the incredible power of comparative studies. You see that in the entire history of science. We certainly experienced that in biology when people in the 1800s began to travel to other continents out of Europe and out of the United States and begin to see some of the similar phenomena that they had observed, but with vast differences. And then do that compare and contrast thing that we all had to do in middle school for essays, but to do it in a scientifically rigorous way. And we have seen and lived through the planetary science revolution in that regard. When I was a kid and even a young college student, we knew so much less about the other planets, and our interpretation of Earth was colored by the fact that we were isolated from these other examples. And so this ability to extend ourselves into the rest of the solar system has opened our eyes in a way that could never have been opened any other way. And so now we understand Earth way better. And that's not just a sales pitch when you're trying to get funding. It's actually the truth. So we start by trying to know about life on Earth, and that's biology and all of its many different facets, and understand the conditions on other planets, both here and increasingly exoplanets. And then we try to map the life stuff onto the planet stuff. And so it seems like 3 easy steps, and of course none of them are easy. And this last step of mapping the life stuff onto planet stuff is very, very difficult because studying the planets themselves in the spatial excuse me, and temporal scales that we need in order to seek evidences of some sort of lifelike process is much more demanding than characterizing a lot of the other aspects of planetary science, although the other aspects of planetary science have greatly informed that. So on Earth, you know, biosignatures, we're kind of surrounded by them. And if you ask the typical 4th grader, going back to 4th graders, or maybe even 2nd graders, what you know, a sign of life on another planet would be, they might come up with a big dinosaur bone. And dinosaur bones are wonderful. I love dinosaurs, and I love big fossils, and have helped put fossils together and all that stuff. I mean, it's really compelling. But because we are anticipating that the most likely pathway for life to arise and prosper on other planets comes from a microbial foundation, just like it has done on Earth, then we have to look for much more obscure and nuanced evidences. And so the image on the right is not so obvious. That is an iron coating within which there are many, many iron oxidizing bacteria, but when you look at it, it just looks like minerals. And so you have to work very hard to find out that there are actually organisms in there that are helping oxidize that iron. And so the suite of questions that we are faced with is inherently multi-dimensional in that we need many lines of evidence to demonstrate that something is alive, compared to watching a giraffe walk in front of you, where you have a pretty good intuitive idea that something that is large and moving is living. So the biosignature categories, for those of you who are not steeped into the field, I want to just go through those just briefly to tie this part together. We know that the complexity of our atmosphere and the constituents of our atmosphere on this planet are highly chemically incompatible over short timescales. And so on our planet, we know that this is attributable to life processes constantly uptaking and giving out a lot of these gases and churning on extremely short timescales when you think of it in the geological context. The search for biological molecules, from the point of view of exoplanets, there's the suggestion that we may be able to see planet-dominating proteins like chlorophyll that are light-capturing pigments, and maybe others that have been optimized for other solar spectra around other stars. And so the top component, the atmospheric components, and this kind of globally dominating potential for some kind of biological polymer that can be detected are really our great hopes for understanding the potential biospheres on exoplanets. When we are looking at our own solar system, we have the potential for actually landing and getting samples, studying them in situ, or for sample return missions, the era of which we're only just beginning to embark on now. And bringing these materials back to Earth or studying them on the planet of origin and trying to look for what we would call biomolecular fossils of some sort. And this field has really, on Earth, exploded in the last 25 years. The ability to find, detect, and characterize ancient remnants of life that are hundreds of millions of years old, and in many cases billions of years old, within rock materials is something that I don't think we suspected. Perhaps 50 years ago, that you could look in an ancient chert and find distinctive lipids that only come from living things as far as we know, and that can be tied to particular groups of organisms. For example, hopanes in sponges have now pushed back the sponge fossil record from maybe body fossils in the 500 to 600 million year range back to at least 850 million years. So even though we don't have chunks of fossils that look like ancient sponges from that age, we have their traces. And so that certainly has infused the field with great enthusiasm for looking at that. Of course, we are looking in a lot of our life detection efforts within NASA and elsewhere at biopolymers and variants of biopolymers and how we detect those, and even elements, so distinctive patterns of elements that would appear only in life processes in the relative proportions that they do, may be a signal. And so we're trying to develop all of that chemical expertise. One of the areas in which we have a lot of difficulty, because it's very hard to do, and that's morphology. So on Earth, it's not that hard to do. You can dig stuff up and you can look at lots of material, and you can find true fossils, both dinosaur bones and microfossils. We can also look for and produce in our laboratories biominerals that we think are either exclusively produced in the presence of life or at least greatly encouraged in the presence of life, biotextures and biopatterns. Obviously, in our solar system, we have not ruled out looking for living microorganisms, although this is very challenging, and we're embarking on that now. Living dinosaurs, I don't know. I hope that they're on some planet somewhere else. And everybody wants to find Baby Yoda, and so do This is just a simplified multiple spectrum of some of the dominant gases in our atmosphere, and I just bring your attention to the very bottom in purple. That complexity of that combined spectrum shows that life is here because it is exceedingly complex. And if you compare that to the atmospheric spectra of the Martian atmosphere or the Venusian atmosphere, you see that they are much cleaner. They are apparently much less complex. And so that feeds into the idea that the atmosphere is a very major thing. Well, for places that we can reach, what do we look for? We all know that microorganisms are tiny, and I think we often lapse into this notion that we're going to somehow stumble upon a cell in a sample. At least on Earth, organisms don't live like that. They live in big groups, and every picture that you see here is an example of that. The Sudan mine is, all of that looks like a rocky carbonate structure that is formed in a typical cave, but actually it's iron oxide byproducts from microbial activity that mimic those shapes. I could go through this entire slide one by one, but I won't. The point is that we know on Earth we can find residual mineral transformations of organisms, even at such times as the organisms themselves have long departed the scene. And we can study those and we can uncover indicators that that was indeed the product of interaction between the mineralogy of the environment and the organisms. Sometimes you're not so lucky, though. So the background picture there was taken 30 years ago now, and it was one of the first pictures taken when we broke through into a very famous cave, Lechuguilla, in southern New Mexico. It was a tiny little pit, and through the efforts of many cavers, it was dug into, and it's become one of the largest caves in the world. It's the deepest cave in North America. It's 150 miles of map passage and still going. And it's a wonderland of microbial mineral interactions. But we studied these particular features early on because they looked so biological. And I got to tell you, there was nothing in there. There was no signal of organic carbon. There was no microfossils. There was nothing organic of any sort that we could extract for it. And so all we had was the morphology. It wasn't until we had the good luck to be able to study other features like the pool fingers that you see on the bottom. You can see the pool line there. These are formed and they are carbonate, but if you slice through them, as you can see in that cross section, they are banded, and they are banded by biological and non-biological episodes in their history. And so we can actually see that some of these features do have and can preserve some of those evidences. Going back to this previous slide, the gooey, drippy thing on the upper right-hand side that we call a snotite for obvious reasons, when we found those living in a cave, that enabled us to complete this likely causative cycle. And so now we believe that these objects were, in the history of this particular cave, were one-time microbial things like the snotites, and that they were replaced by minerals over time, which is a common process in mineralogy. Biominerals, in general, have garnered a lot of attention, and a lot of this has been due to the exemplary work of Bob Hazen and his colleagues. And this is something that I latched on to the minute he published it, because it was a wonderful way to hang everything together for teaching. And their notion is that we can see the degree of life's influence on our planet by looking at the suite of minerals that we have. And we have a fabulously rich mineral array on Earth, and a lot of them are in my mineral cabinets in my house, just like everyone who loves minerals. And their estimate of 4,400, 4,500, about 1,500 of those they attribute to the presence of the fact that we have two very important things on our planet that also probably contribute to life, and one is abundant water in the liquid form and tectonic mixing. So we have a lot of recycling mechanisms on Earth. But the rest they attribute to life, the presence of life and all of the chemical processes processes that life undergoes. And certainly the organisms that we study in the geomicrobiology field are right there with pedal to the metal, literally, because they are transforming minerals. So this is another huge idea, I think, that has come into our thinking, and Bob and his colleagues have put it into focus for us. Well, we can reproduce these in the lab. Okay, this is just some of my favorite pictures from some of my favorite organisms. These come from caves. The lower left-hand image is a naturally occurring sample. All those beautiful little stars in there. Inside each one of those little stars is a little cell, a little tiny, tiny, tiny cell. And it took us years to convince ourselves that these were actually organisms. By the time they've produced the manganese stars, manganese oxide stars, they've essentially sealed themselves in and killed themselves, but they manage to perpetuate themselves in many, many rocky environments. And so we have reproduced a lot of this crystal formation. The other two images show laboratory syntheses of organisms in the presence of precursor, highly reduced minerals and the transformation of those into oxides. And this can occur even in anaerobic conditions because many microorganisms can use non-free oxygen as oxidants. And of course, this is something that is of great discussion when we look at other bodies in our solar system and we're trying to figure out how are we going to look for potential chemical and other signs of life. In the subsurface, we have a lot of elaborate living morphologies on the microbial scale. And I point this out because, you know, for a long time, the joke in microbiology has been, how many organisms do you have? And the joke answer is 3. You have spheres, you have rods, and you have spirals. Okay? And if you look at a lot of the microorganisms on the surface, they kind of do fall in that. I mean, if we took a swab of your teeth bacteria right now and we actually looked at them, we would see a preponderance of those shapes. But in places that are different, that are hidden away, the potential for complicated shapes is vast. This must mean something. And originally, our hypothesis was that the lack of predators, the lack of grazers in a lot of these chemically very extreme and thermally very extreme environments that we were studying must mean that things could be as funny shaped as they want. Well, that was an okay hypothesis, very hard to demonstrate one way or another. It occurred to us later that actually that must be meaningful in a more deep way. And this has still not been adequately tested in any way that I understand. If anybody's working on this, I would love to hear about it. But the manipulation of the microphysics of a microorganism's environment is of tremendous benefit from that organism if it can do that. An organism of the size of a microorganism, 1 micrometer plus or minus in diameter, experiences the universe in an entirely different way than we do. You can get punched around by Brownian motion. Viscosity has a whole different meaning for you. Your interactions with both the chemistry and the physics of your environment is on a scale that we have a great deal of difficulty thinking about because it's so extraordinary from our perspective. Here's just a few more of my favorite shapes. They come in just about everything. And we see these over and over again, and we're beginning to map out where we see different morphologies. Many of the organisms that share a morphology do not share a close genetic history. So this means that the shapes must be functionally adapting them in some way that we don't yet understand. One of the things that bedevil us, I believe, not only on Earth as we try to do our field studies and try to interrogate nature here, but also as we try to do the even more difficult planning for future space missions, and that is, what spatial scales do we integrate? And this is, of course, one of our greatest eye candy areas that our team has ever worked in, and this is the NICA system in Chihuahua in Mexico. It has been the subject of several National Geographic expeditions and TV specials and blah, blah, blah. We are potentially thinking about a third. And in this particular environment, of course, you have these spectacular crystals. They're some of the biggest in the world. We looked inside them. We found organisms. We found DNA and all of that stuff. But the question is, what would you do if you were a robotic mission trying to figure out what you take along? Do you take along cameras? Do you take along microscopes? I think in this case, it would have been very hard to demonstrate without visual evidence that there was something there to study. So this is something to ponder. How do we go forward and do this thing that is in many ways harder than chemistry? Because at least the way we do imaging on Earth at the microscopic scale is more labor intensive. Does it have to be? Maybe not. And there are people within NASA and within the university and private sector who are working on ways to try to do this imaging. And I think this is a very important component. So one of the heroes, of course, of the entire story is the entire group of extremophiles, as they're called here on Earth, just meaning that if we think it's a tough environment, then we kind of slap the extremophile label on them. Some of them are more extremophile than others, and some of them are just extremotolerant. So whether they actually love where they're living or whether they're just putting up with it can be a subject for debate. One of the things that I presented at the Library of Congress on a recent talk that I actually shoved in here just last night, and that is the issue that, we know a lot about them. We know a lot about them in terms of their genetics. We know less about them in many ways about their physiology and how they actually operate together and operate in nature. And so I just picked out a few things that I think are a big deal that we wrestle with over and over again. Those of you who have sat on astrobiology panels or working groups or study groups or National Academy studies have probably come up against these, and one of them is the minimum water activity. We have ideas about what we think the minimum water activity can be, but people keep popping up with potentially contrary evidence, but it's very, very hard to understand, and we do not have a rigorous understanding of whether or not we can support organisms on vapor only. Does that happen here on Earth? I kind of think yes, but thinking yes kinda doesn't mean you're demonstrating it, right? And so this has profound implications for how we go out looking for life forms elsewhere. I already mentioned the manipulation of the surface microphysics. The whole arena of multiple tolerances and synergy, whether that synergy is positive or negative, in terms of suites of characteristics that extreme organisms have that they bring to these multifaceted environments in which they live. Very often you have very hot or very cold, very salty places. How does the salt tolerance and the temperature tolerance interact? Do they help the organism? Do they hinder it? Are the genetic components that they use to meet these challenges the same? We don't know, because it's really hard to study. And then the degree to which organisms can kind of sit and twiddle their thumbs, so to speak. We have a classic idea of dormancy, where organisms shut down. And then we have the other opposite part of that, which is where they're highly active. But what about the point at which they're malingering, okay? Where they're not fully active, maybe they're not active at all, and yet they're not really burning energy. We used to think that they all burned energy at a certain rate, and we used to call this, the minimum maintenance energy and stuff. I've stopped believing in that. I've stopped believing in that because of the things that we are finding in nature, the things that other folks who are working in this field are finding in nature that don't really fit that. So this is a conundrum that we must solve. So of these biosignatures that I've talked about, a lot of them are highly case dependent. If we're looking for polymers, of course, they're also they're chemistry dependent, right? If we're looking for lipids, we're looking for lipids. We're looking for DNA or alternate DNA, we're looking for the chemistry. Is there a way that we can float above that? And I hate to say it, but I think physics is the way to float above that. Just to make physicists even more sure that they are the real science. Our missions to Mars, in the future Europa, thoughts about sending missions to Enceladus, our new mission Dragonfly, which is going to Titan, about which I'm exceedingly excited, and even the role of small bodies and minor planets, and our evil twin sister Venus. These are all of extreme astrobiological significance. Venus, for example. You know, people have often said to me, well, Venus, that's not really on your dance card because, Venus, people have suggested that maybe there are some kind of life forms in the, 50-kilometer and above cloud deck, maybe. I think that's worth checking out. But there's another deeper, more philosophical comparison here, and that is Venus is so similar to Earth in so many ways and yet so different. And Venus must have had horrible things happen to it in its history. Did it have a biosphere before then? Is there any possible way we can tease that out? This is a question that I think we're only beginning to be able to think that we might be able to answer. And we don't have the way to answer that yet, but it's worth thinking. And then I had the privilege of being on a paper with Dale Cruikshank and Yvonne Pendleton and a bunch of the Pluto folks. And who cannot love Pluto, right? I mean, Pluto is just one of my favorite little guys. And the amazing amount of complex organic chemistry and other chemistry and complex geology on that little body has put it into a different realm. I think it is now of astrobiological significance because it is a potential home. Maybe it's not a home now. Maybe it'll be a home when the sun becomes a red giant for a while. Who knows? I think that, freeing ourselves from the very simple-minded origins of the field of astrobiology, which were hard enough, and trying to have this solar system-wide, galaxy-wide view is something that we're on the precipice of. I want to show a couple of slides that I have shown a lot. So those of you who've seen me talk before have seen this. We have focused heavily in the early part of astrobiology's history and exobiology on our own type of planet, our own biosphere. This makes a great deal of sense for exoplanets because of what I talked about before, needing atmospheric signals and things like that. It has to be a big, surface-driven, energy from the sun, globally dominating biosphere. But when we look in our own solar system, we've got a whole different set of objects that we're interrogating, and all of them fall currently into this category, which is no visible means of support on the outside, meaning they're not obviously photosynthetic. None of them are green, none of them are gooey. They either have no atmosphere or a thin atmosphere. If there is life there, it has to be fed by geochemical sources. The energy has to be coming from the interior of the body, not from solar-driven circumstances. On Mars, you could make the case that the surface obviously receives enough light, but what about the icy moons of the outer planets? There is just not enough sunlight to run anything much. And there have been papers produced even in the late 70s, early 80s, really doing those calculations. So where do we go from here? And why am I predicting the future? It has been pointed out to me that I share several characteristics with a certain other person. Not just the glasses and the fuzzy hair, but also the fancifulness. But, I've been doing this for 40 years plus, and I'm waxing philosophical in my old age, which many scientists do. It's a real hazard of growing old. You think you know more than you do. But I do think that this is the seminal question. I know we're looking for the origin, and we've got the classic 3 questions that we have in NASA, but really, to me, they all boil down to this: where is there a home for life? Is there a home for life on Earth? Well, we know yes, so then we go back in time and interrogate, how did that happen? Is there a home for life on Mars? Was there a home for life on Mars? Was there a home for life on Venus? Is there a home for life now in the icy moons? The search for looking for that abode and looking for potential inhabitants is it. This brings up a sticky problem, and that is, what if it's not like our life? People talk about this all the time. And every public talk, I'm sure any one of you who are in astrobiology have ever given, you get a question from the audience about what if it's built out of silicone, or what if it's, because these ideas are out there, they're out in science fiction movies and books and all of this stuff, and so people know that there's this possibility, and we kind of wave it away and say, well, what we know now is what we have to base the science on, and that's true. But how do we cope with this? It may be that organic goo-based life is rare. I hope that's not true, but it might be true. And so we have to try to prepare as much as we can. And of course, work by people like Steve Benner and other folks working on weird life, as we call it, is invaluable to this mix. And in fact, the work of synthetic biology is invaluable to this mix because maybe we can make weird stuff in the labs here on Earth that we would never dream up and not recognize. And maybe we'll find it somewhere. So astrobiology is advancing rapidly along a lot of other fronts, but hardly at all along others. This is probably natural for the cadence of individual subfields, but I think that those of us who are thinking about how we go forward through this century and into the next in terms of our exploration for life and our characterization, hopefully, of organisms or life processes elsewhere, have to think about keeping a balance within all of the different areas of astrobiology. We certainly are very much on track to try to make the areas of astrobiology that we interrogate mean something for our interpretation of real data on other bodies. And so, unlike NSF, which funds basic research for its own sake in many cases, certainly our science within NASA itself is colored by this, this drive to create and make missions. One of the solutions to this has really dropped out of my own work with my team and various colleagues over the years, and this is that in spite of the differences in chemistry that we've seen in subsurface environments all around the world in all different temperatures, all different degrees of acidity and alkalinity and saltiness and not saltiness, and et cetera, we see the same kinds of ecological behaviors. If there are universal signatures, they may not be things, they may be clues as to behavior, and that behavior is driven by the need for energy and by the need to sequester materials and by the need to get rid of waste materials and so forth. And so if there is hope for us to actually expand our consciousness into this arena of universalizing what we know from Earth and take that elsewhere, I think it lay in trying to extract from the physical things that we study, the objects, the organisms, the chemistry, what exactly are we going for. Biopatterning is one of those examples. I don't have any time to explain this in any detail, but all of those fancy hieroglyphic-looking maze-like patterns that you see there are the interaction of myriad microorganisms of many different kinds with certain characteristics that all come together to reproduce these patterns, no matter what the organisms are. We even see large-scale reproductions of these patterns in desert vegetation, in desert soils. All right, so there's something about these patterns that really matters. We have a number of papers out on the mathematical reproduction of these. We're trying to interrogate what they actually mean. So this is part of that search for deeper meaning. So we must seek general principles and universal explanations, but the problem is we have to have them testable and implementable. And that's hard enough on Earth, and it's very challenging when we try to apply this to other bodies. I think I'm going to pass that slide over and pass that slide over because I have 4 seconds left. I don't know how many people are reading Strange Planet by Nathan Pyle. Oh my God, I love that cartoon. But this is one of my favorite little clips from one of his 4 panels. It's sometimes science is so difficult it makes me sad. And that's true, but on the other hand, the rewards are very great. And in spite of the fact that I may drop dead one of these days, before we have actually discovered life on another planet in our solar system or beyond, I think that I'll still be okay with that. Although what I really secretly want to find is this. Okay, thank you very much.
Thank you very much, Tony. That was, sorry, Penny. That was fantastic. I think you're going to have to shout your questions, but we do have some time for questions. Yes, please.
Liquid or vapor water is necessary for life to arise?
The question is, do I think liquid or vaporous water is necessary for life to arise? If it's based on organic chemistry, then that's one of the best bets in town. If you're looking at a very different temperature regime on a planet, you may have other solvents, and people have suggested those. Oh, Pluto. I don't know. We should go with more missions and figure it out, don't you think? Okay. I can't see anything.
Yes, it's hard to see here, but any other questions?
Yeah, yell it out.
Yeah, are you familiar with the proposed Finesse spacecraft? Finesse?
Finesse, yes.
Yeah, fast infrared exoplanet spectroscopic survey explorer.
Yeah.
Proposed for, I think, a 2022 launch. Do you have an opinion on how much you think this will advance our capability to detect and identify exoplanet life?
I'm a NASA person, and I can't endorse a mission unless it's one that I'm pushing myself and I'm on the team. So, I don't know. I can't really comment on that. Yes, please.
A long time ago, I read James Lovelock's book, Gaia, and he said that the signature of life was when entropy was uncharacteristically shifting the processes. Is that still valid, a valid test?
Yeah, the question is about whether or not the Lovelock and Margulis's Gaian ideas, which includes a departure from equilibrium and entropy and all of that, was still valid. I think it's very valid. The idea that you have to be wildly out of equilibrium has been challenged by mathematicians, by a guy who does mathematics and origami, which is really interesting, looking at what is the optimum entropic state for a complex system like life to actually get hold and function. So I think there's a lot of thinking about that. A lot of it, of course, is still entirely in the theoretical realm, and how do we then translate that and actually test those ideas? Over the 30-something years that the Gaian ideas have been published, which influenced a lot of my thinking early on, it's proven to be exceedingly difficult to conduct rigorous tests of those principles because we haven't had the tools. But I think that algorithmically we are advancing tremendously, and perhaps we will have the computational tools to really test, at least in the model world, some of those ideas.
We have one more question.
Okay, one more question. Yeah, Alan. Hi.
Thank you for a spectacular Sagan lecture today.
Thank you.
When you look back on your career so far, what would you characterize as the one or two most significant findings related to our understanding of astrobiology during your career?
Oh, Alan is asking what are the one or two most significant findings related to astrobiology. Well, so I have a certain bias, okay? I think that when we first started, we were looking at the surface of the Earth and in the oceans. And now we understand that a huge part of the shallow crust is inhabited by organisms, and in fact, the biodiversity probably exceeds all of the biodiversity on the surface. And the reasons for that, we hypothesize, are a lot more partitioning of the subsurface because it's a solid material with liquids flowing through it. So I think that has opened our eyes to the fact that the planet that we thought we were living on has a whole hidden compartment that is perhaps as significant, maybe even more significant, and maybe even allowed life to survive various catastrophic events on the surface. The ability to interrogate the genetic material. I mean, I don't know how you beat that. We can now read the secret code of who interacted with who and who's related to who, and maybe try to eventually figure out how they got there. It's premature. We have such a tiny window on the organisms that are out there. So my mental picture of the biological world, particularly the microbial world, is this giant point cloud of information. And they're temporarily packaged in organismic bits and community bits. But the flow of information is so fluid, as we now understand it, that really is revolutionizing our grasp of evolutionary processes. We're not good at it yet, but I think we're, coming upon the era where people are paying more and more attention to that. So that's a sleeper. Thanks.
Thank you so much, Penny. Let me present you this.